USDA logo, United States Department of Agriculture.
Hi, we'll go ahead and start the program. Hello, everyone. Welcome to our Food Safety Conference and the Stakeholder Workshop. I'm Sunny Noh. I'm the Project Director for a multi-state and a transdisciplinary program funded by USDA NIFA. Our main goal is to address food safety issues during fresh and fresh-cut produce, washing, packing, and retail display. Before we start, I would like to recognize our project team, our talented research scientists, engineers, and economists. You all did a great job in developing solutions to the food safety challenges and preparing this workshop. I want to thank our postdocs, students, and the support staff. You are the star and our future. I'm especially thankful to our stakeholder advisors. On behalf of the entire project team, I want to thank you for your great guidance and support. I want to thank our industry collaborators. Thank you for providing countless support to our research and opening doors for our team to conduct studies under the real-world conditions. I'm grateful to our project sponsor, USDA NIFA, the participating institutions, and collaborating universities. Without the funding and your strong support, this project would not have been possible. The workshop has 6 sessions covering from packing house operations to retail, from fundamental research to applied studies. During the workshop, your mics will be muted. You are encouraged to provide feedback and your questions via the chat. Without further ado, let's start our session 1. Improving food safety of leafy greens, tomatoes, and papayas during fresh cut and packing house wash operations. Thank you.
Good morning. I'm Pat Milner. I'll be the moderator of this session. Just want to remind you of a couple of things. During the sessions, we will save all the questions via the chat, so you're encouraged to use the chat for any questions, and they will be at the end of the whole session. You may need, for some of the presentations, to increase the volume if you have a low audio sound. So you will be able to control your own volume on that. I'm the first speaker, so I will begin my talk now. This presentation focuses on studies we conducted to address the potential for cross-contamination by salmonella on tomatoes when free chlorine sanitizer is used in dump tanks and plant debris enters the tanks along with the tomatoes. The problem our team addressed centered on the dump tank inputs from field harvests, the decrease in water quality from these inputs, which led to rapidly changing conditions in the dump tanks relative to free chlorine oxidation, reduction potential, pH, temperature, and the need to measure them along with measuring the microbial content. Our team first established a procedure to simulate dump tank water quality in our lab studies. Cherry tomatoes direct from a supplier's harvest were delivered to our lab in Beltsville. The team conducted a series of chlorine dosing experiments to assess the impact of increasing free chlorine concentration on microbial survival and mesh filtration to eliminate particulates greater than 300 microns to which microbes may adhere. The final tomato water procedure used to prepare water in subsequent studies is shown at the bottom of this slide. Here we see that the initial wash water chlorine treatment yielded a post-chlorination 4-log reduction in all 3 microbial groups, with or without 300 micron filtration. At 150 parts per million free chlorine, all 3 microbial groups were reduced to approximately 1 colony forming unit per 100 mils in wash water when filtered, the blue bars, but not as much when unfiltered, the orange bars. These graphs show that even after 150 parts per million free chlorine dosing and 300 micron mesh filtration, tomato fruit microbial counts for these 3 groups of microbes were only reduced approximately 1 log. The next series of experiments used simulated tomato dump tank water to examine the separate effects of debris and tomato fruits as sources of cross-contamination by indelibly marking some fruits to be inoculated with a cocktail of kanamycin-resistant salmonella and commingling them in simulated water with uninoculated fruits, then determining cross-contamination on the fruits and survival in the wash water. In a separate inoculation challenge, tomato debris that had been separated from the harvested mix was inoculated with a cocktail of rifampicin-resistant salmonella and commingled with uninoculated tomatoes to assess debris as a potential source of contamination. Salmonella in filtered and unfiltered wash water with debris was also assayed. For the 3 separate trials conducted, 2 used cherry tomatoes, 1 used grape tomatoes. The strains of salmonella used to inoculate the tomatoes and debris are distinguished by their antibiotic resistance factors, as shown here. Trials 2 and 3 also included high and low salmonella inocula concentrations. This table shows the wash water parameters for each trial set. pH was maintained at 6.5 for all target-free chlorine dosages. Pre- and post-wash free chlorine measurements show that the target free chlorine levels were mainly well achieved. TDS, COD, and chlorine demand were measured for each trial. This graph shows the log reduction in salmonella on tomatoes and debris at 0, 50, and 150 milligrams per liter free chlorine. These results are consistent with previous reports of chlorine effectiveness in washing tomatoes and other fresh produce. The reduction of salmonella is comparable to that of APC, which is not shown here. Here, the average MPN estimated salmonella recovered from 3 trials of uninoculated tomatoes commingled with high and low dosed inoculated tomatoes were compared to trials with inoculated debris. The scale here doesn't show, but there was one cross-contamination event detected at 0.04 mpn per gram with 150 milligram per liter free chlorine wash. Harvest debris in this set was a greater source of contamination than that from the inoculated tomatoes. This graph shows the percentage of uninoculated tomatoes from which salmonella were recovered after commingling with either inoculated tomatoes or inoculated harvest debris at 3 doses of free chlorine, 0, 50, and 150 milligrams per liter. This table shows salmonella originating from harvest debris survived better in unchlorinated water than salmonella originating from tomatoes. In summary, chlorine washing significantly reduced APC and salmonella on tomato fruits, but was not significantly effective in reducing APC and salmonella from field debris. There was no evidence of salmonella survival in water in the presence of free chlorine. Cross-contamination of salmonella was sporadically observed at very low frequency at all free chlorine levels tested. These findings help us learn that field debris might be a more important target for reducing the risk of foodborne pathogens, and that surface contact might be a more plausible route of cross-contamination than that mediated by wash water. Next, we examine microparticulates from tomatoes and debris as a source of wash water contamination using the differential antibiotic resistant strains shown previously. This graph shows salmonella counts in tomato wash water after filtration for 3 pore sizes compared to unfiltered water for 6 trials. Results clearly show the harvest debris, even at 100 milligrams per liter free chlorine, the light green bars, was the main source of salmonella contamination in the presence of free chlorine. Virtually no counts from the tomato fruits, the orange bars, which are not visible because that's the blank spaces in between the dark green and light green, at 100 milligrams per liter chlorine. The 63 micron mesh filtration eliminated 93% of the particulate-associated salmonella contamination. Wash water conditions here were as shown. The size distribution of particulates based on the percent of total surface area by size range is shown for unfiltered tomato wash water in the graph on the left, and for samples filtered through the 3 different mesh sizes. The photomicrograph on the right shows the diversity of particulate shapes and sizes in the tomato wash water, potentially serving as harborage sites for salmonella and other bacteria. Particulate size significantly affected the microbial community. After chlorinated wash, removing particulates larger than 330 microns and especially larger than 63 microns reduced the presence of gram-negative bacteria, and this selectively increased the percentage of gram-positive bacteria and spore formers such as Terribacillus and Bacillus in the surviving microbial community. This effect was more pronounced for water collected from a commercial packing facility, probably because repeated addition of chlorine during operations. Susceptibility of gram-negative bacteria and resistance of gram-positive bacteria to chlorine may have contributed to this differential survival. In summary, No salmonella originating from inoculated tomatoes survived in the wash water. Salmonella originating from debris, on the other hand, did survive, and the bioburden was greater on larger size particulates. 93% of surviving salmonella were associated with particulates larger than 63 microns. Cross-contamination from debris to tomatoes was much more frequent than transfer from tomato to tomato. The research presented in this session represents the collective efforts of our ARS team and university cooperator, as well as our industry partners, United Fresh, Littman Produce, and other tomato producers, along with NEFA's grant support. I invite you to read the two publications which provide details of this research. They are available through the DOI numbers shown below. Thank you for your participation.
The next speaker is Dr. Sunny Lowe, and she will talk to us about packing house operating conditions impacting human and plant pathogen infiltration into tomatoes.
Packing house operating conditions and the infiltration of human and plant pathogens and the development of market disease. Why do we need to study both human pathogen and plant pathogen? Well, packing house operating conditions such as dump tank temperature differential and tomato immersion time affect both human pathogen and plant pathogen infiltration, and thus food safety and market disease soft rot. While there have been numerous studies separately evaluated human pathogen, Salmonella, and plant pathogen, Pectobacterium, or previously known as Erwinia, or ECC, in a single setting. For risk assessment and mitigation, we needed to know, does dump tank temperature differential between water and tomato emerging time affect both pathogens equally? Do tomato varieties respond to pathogen infiltration equally? And also, how do pathogens get inside and where would they go once internalized? Many questions. The objectives of our study are to address those major data gaps identified. Testing conditions. For Erwinia or ECC, it was isolated from a decayed tomato showing soft rot symptom. It was then transformed for GFP and antibiotic resistance marker for infiltration studies. Then we used the wild type for tomato soft rot development. For temperature differential, 0 degree differential means tomato and wash water has the same temperature, and 10 degree Fahrenheit positive temperature differential means wash water is 10 degrees warmer than tomato, and vice versa. Our first step was to see where bacteria are located once internalized. As you can see, bacteria do not randomly go everywhere. Most internal tissues are actually pretty sterile, except this central core area, where the vascular bundles connect the stem scars and allow air and water go in and out under certain conditions. That was with artificially inoculated salmonella. For naturally contaminated bacteria, the same trend is true. Our follow-up studies focused on exclusively the stem scar and the central core tissues. For pathogen infiltration, we considered both the frequency or the incidence of contamination and the bacterial populations inside tomatoes. As you can see, there is a significant interaction between immersion time and the temperature differential. Increasing immersion time or negative temperature differential significantly increased salmonella infiltration, with the worst case scenario happened when both are increased. A positive 10-degree temperature differential, however, did not show appreciable benefit in reducing salmonella infiltration, but a negative temperature differential certainly pushed the pathogens inside the tomatoes. We also observed substantial varietal difference in salmonella infiltration in response to temperature differential and immersion time. Under the exact same testing condition, variety Sunbright showed substantially more pathogen infiltration than variety BHN. Immersion time and the temperature differential also significantly impact how far does bacteria travel inside the tomatoes. With a negative 10-degree temperature differential, salmonella cells are mostly around the stem scars and they immediately underneath the scar. However, under the same temperature differential, for 15 minutes, salmonella cells can travel down to 25 millimeters below the scar. Now let's look at the infiltration of both salmonella and Auvernia in tomatoes. Very interestingly, they actually showed a very similar response to temperature differential and immersion time. Figures on the left are for the 2 minutes immersion time. Figures on the right are for the 15 minutes immersion time. And you can see side-by-side comparison, they behaved very similarly, which is good news, which means that the packing house operations don't have to do things differently to control both. Now, let's compare the infiltration of salmonella and soft rot development in their response to temperature differential and immersion time. Both salmonella and soft rot increased with the increase in negative temperature differential and immersion time. However, for 2 minute immersion time, soft rot was minimal at all temperature differentials, whereas salmonella infiltration was substantial at 30-degree and 10-degree negative temperature differentials. You may be wondering why. Well, soft rot development is highly dependent on the population of ECC internalized. To develop a substantial amount of soft rot, you need to have at least 100 cells per fruit. And this is true for both varieties of BHN and Sunbright. In a separate study, we evaluated the pathogen infiltration pathways. On your left are optical images. It shows the penetration of dye through stem scar and vascular bundles. On your right are micro-CT scan images. Because tomatoes do not have bones or contrast inside, we let potassium ion dye, the contrast agent, infiltrated in tomatoes naturally, so it illuminates the passage of the chemical through the vascular bundle. We also used laser confocal microscopy to see the movement of fluorescent beads of bacteriocytes and the GFP-tagged bacteria. As you can see here, not only that bacteria and the beads moved through the vascular bundle, but you can also see the xylem tubes of the vascular bundles played an important role in the movement of bacteria and the beads inside the tomatoes. I don't want you to think vascular bundle is the only pathway for bacterial infiltration. When the stem scar is damaged, like this one on the right, the dye can move right in without bothering anything else. So it is very important to avoid damage to stem scars during harvesting. In summary, internalized pathogens are mostly located in the stem scar and the core tissues underneath the scar. Salmonella and Aeruginosa infiltration in tomatoes showed similar response to temperature differential and immersion time. However, soft rot showed both similar and differential responses. Although we did not see a huge benefit by increasing the positive temperature differential, however, increasing the negative temperature differential, especially when the water is colder than tomatoes, you will certainly increase the risk of both Salmonella and Aeruginosa infiltration as well as soft rot disease development. Increasing immersion time not only increases the risk of Salmonella and Aeruginosa infiltration, it also increases the populations and the depths of infiltration of bacteria inside the tomato core tissues, as well as soft rot. Those are the publications cited, and they are available at our website. I want to thank my team members, collaborators, especially our industry collaborators in this study. Last but not the least, thank you very much.
Our next speaker will be Dr. Shen Wu. He will talk about the evaluation of Salmonella cross-contamination associated with cleaning tools for papaya washing.
Thank you. Now we switch to our work on Salmonella contamination on papaya. Papaya is not in our NIFA proposal. Here I briefly explain why we conducted this research. US is one of the largest papaya consuming countries and the largest papaya importer. We consume about 50% of papaya on the international export market, and 75% of US imports comes from Mexico. So Mexican papaya is a vital commodity for US fresh produce supply. On the other hand, Salmonella outbreaks associated with the consumption of papayas imported from Mexico has been frequently reported. There has been 8 since 2011, according to FDA. In late August last year, FDA issued a statement to all sectors of papaya industry, calling for the concerted effort to improve Mexican papaya safety. It contained a 4-point call to action, including root cause analysis of contamination sources, agriculture and wastewater risk analysis, and increased research activities. United Fresh Produce Association and the Texas International Fresh Produce Association lead this effort by developing a new industry guideline, Food Safety Best Practice Guide for the growing and handling of Mexican papaya. However, there is very limited data regarding Salmonella on whole papaya fruit and contamination to support this great effort. So, per request from the industry, with the approval of NIFA, we conducted this research to provide information regarding the data gaps. One of the key data gaps is regarding the risk associated with the use of sponge or other fibrous cleaning tools. Mexican papayas are typically hand harvested and washed to remove dirt and latex before packing. Wash water containing 100 to 150 ppm free chlorine or 80 ppm peracetic acid is typically used. Both single and double flume washing systems are common. Typically, workers use sponge or wash meats to clean each papaya to remove visible dirt or latex. In some cases, the sponge is used for the whole shift. The main objective of this study was to assess the risk of washing tools, including sponge and microfiber meat, serving as vectors of Salmonella cross-contamination during post-harvest washing. Therefore, we wanted to examine how well Salmonella can survive and transfer in a simulated washing system. Due to limit in resource and time, the experiments were conducted by benchtop washing of individual fruits. Salmonella strains with a resistance to rifampicin and to calamicin were used to differentially inoculate papayas and sponges. The concentration of the Salmonella inoculation were very high to create a situation that would be worse than the worst case scenario of naturally occurring contamination. Wash water was generated by soaking papaya fruits with dense surface incisions to release latex. Free chlorine up to 100 ppm and PA up to 80 ppm were tested. We used pieces of sponge and microfiber meat as wiping tools. From now on, when we discuss the result, sponge often means both sponge and microfiber meat. The experiments were simple. First, papaya fruit was inoculated with Salmonella strains resistant to rifampicin. Then, we washed one inoculated and two non-inoculated papaya sequentially in wash water containing different concentrations of chlorine or prussic acid. The sponge was inoculated with Salmonella resistant to kanamycin, wetted in wash water and squeezed dry before wiping the whole surface of the first papaya. And the same sponge was used to wipe the second papaya without wetting and the third papaya after wetting. Each papaya was submerged in wash water for 30 seconds before being wiped with a sponge, and followed by another 30 seconds submersion in wash water. Then survival and transfer of Salmonella on papaya fruit, sponge, and water were determined. Salmonella on papaya was determined by peeling an area of 100 square centimeter, followed by selective plating and overnight enrichment. This is a reduction of Salmonella on inoculated papaya. We can see that the reduction of Salmonella counts on the fruits were limited under all the conditions we tested. Approximately 2 logs when sponge was used and 1 log when wash meat was used. And the reduction of Salmonella was poorly correlated to the concentration of sanitizer used. In fact, the reduction with or without sanitizer was not significantly different. On the other hand, the reduction of Salmonella on the sponge was very high in the presence of any sanitizer. Salmonella was not detectable by selective plating, with the exception in one case where a single colony was detected after washing in 25 ppm free chlorine. However, viable Salmonella cells were detected on sponge after overnight enrichment, as indicated by the stars in this figure. Next, we looked at the transfer of Salmonella inoculated on papaya to sponge and to subsequently washed papaya fruits. Using selective plating, the transfer was only detectable when no sanitizer was added to the wash water, as indicated by the solid circles. However, after an overnight non-selective enrichment, Salmonella transfer to sponge and further to the second papaya fruit was sporadically detected, as indicated by the open circles. Again, the sporadic transfer had a poor correlation to the sanitizer concentration. However, this transfer was eliminated by dipping the sponge in wash water before wiping the next papaya, as shown in panel 3. We further looked at the transfer of Salmonella cells from sponge to papaya surface after inoculating large number of Salmonella directly on the sponge. Again, transfer was not detected by selective plating except in washes without sanitizer, as indicated by the solid circles. And the sporadic transfer was detected after overnight enrichment, as indicated by the open circles. Higher frequency of transfer to the ends, which include the stem and the floropid, was observed. At last, we examined the Salmonella transfer and survival in wash water. Salmonella inoculated either on sponge or papaya was not detected in wash water with any tested sanitizer concentration, either by selective plating or by overnight enrichment. In summary, Salmonella population on papaya was significantly reduced after washing with either chlorine or PAA as a sanitizer. The reduction was not strongly correlated to sanitizer concentration. Salmonella inoculated on sponge or microfiber meat was reduced to undetectable by selective plating with either chlorine or PAA as sanitizer, but not eliminated. Salmonella survival in wash water with either chlorine or PAA as sanitizer was not observed using any detection methods. Salmonella transfer in the presence of sanitizer is highly inefficient. Successful transfer in the presence of any sanitizer was below the limit of detection by selective plating. Although sporadic transfer was detected by enrichment, the risk of sporadic transfer can be mitigated by frequently wetting the sponge in sanitizer solutions. Therefore, our observation do not support the notion that sponge constitute a significant risk for Salmonella transfer during papaya washing. However, cautions must be used when interpreting the result of this study, as many limitations in experiment design need to be considered. At last, we thank our hardworking team for the extra effort to get this done before the lab closed due to the pandemic. And we especially thank Dr. Jennifer McIntyre from United Fresh and Dante Galazzi from Texas Texas International Produce Association for facilitating interactions with the industry. And thank you for spending time attending this conference.
Okay, for our next talk, thank you, Zhangwu. Dr. Sunny Low will be talking next on monitoring pathogen cross-contamination during commercial fresh-cut produce washing processes using indigenous microflora. Sunny.
Monitoring pathogen cross-contamination during commercial fresh-cut produce wash process using indigenous microflora. Our main focus is on the association between bacterial survival and antimicrobial strengths in real time during commercial operation. FSMA preventive controls rule requires processors to evaluate food safety hazards, identify and implement preventive controls, and monitor such controls. For fresh-cut produce, one of the major food safety hazards is pathogen cross-contamination during washing. If not controlled well, one batch of contaminated produce can ruin many happy meals and cause significant public health and economical problems. For fresh-cut leafy greens, the presence of sufficient amount of chlorine or other suitable sanitizers and the proper pH is often considered as the control measure. With chlorine concentration high enough, pathogens washed off from contaminated produce can be killed immediately, preventing cross-contamination. Maintaining high chlorine concentration in the lab and with pure water is really easy. But doing so with fresh-cut leafy greens during commercial operation is very challenging, especially when you are dealing with diced cabbages and shredded iceberg lettuce. Also, it gets harder and harder as the process goes on and organic matter accumulates in the wash water. The chart shown here is the real-time chlorine concentration in a large pilot plant trial at our industry collaborators facility. As you can see, with each addition of chlorine during the process, the free chlorine concentration that we obtained is lower and lower. Therefore, it is critical to identify the minimum effective chlorine concentration to prevent pathogen survival in water and cross-contamination. Talking is easy, but actually finding the free chlorine concentration that is high enough to minimize food safety problems, but low enough for the industry to implement is hard, very hard. In the lab setting, we tested the effect of free chlorine on the survival of E. coli 0157H7 in wash water and cross-contamination. As you can see, no survival was observed when chlorine concentration reached 5 ppm. However, it took 10 ppm to completely avoid pathogen cross-contamination. In a pilot plant study with our industry collaborator, we found that the survival and the cross-contamination of the non-pathogenic E. coli fluctuates during wash process, but correlates very well with the real-time free chlorine concentration. Since chlorine inactivation on pathogen is dose time dependent, the Produce Wash Water Working Group led by the FDA and the industry wanted to know the dose and contact time required to achieve a 5-log reduction of E. coli in wash water. Because conventional research tools could not achieve this, we invented a microfluidic device, as shown on the right. With it, we found that a 5-log reduction can be achieved with 10 ppm free chlorine and a contact time of less than 0.2 seconds. Keep in mind, I'm talking about reduction in water, not on produce. In 2017, the working group published the guidelines to validate control of cross-contamination during washing of fresh-cut leafy greens. Free chlorine concentration at approximately 10 ppm and the pH around 6.5 to 7 were suggested. Please note we used the word suggested rather than required. The reason includes the concerns of chlorine off-gassing and chlorine byproducts, process capability of the industry, and the need for additional validation during commercial processing. How can we validate the effect of chlorine on pathogen cross-contamination during commercial operation? Because there is no kill step, it is impossible to use real pathogens. But then, what other alternatives do we have? Can we use something like lactic acid bacteria, pseudomonas, anything else? After much consideration, we settled on indigenous bacteria. On the plus side, they are naturally occurring, not artificially inoculated. There is no food safety concerns, and they have no negative impact on shelf life. I mean, no additional impact. But on the negative side, they have no markers and therefore are hard to track. There is quite a bit of variation in their background level. Additionally, their susceptibility to free chlorine in comparison on that of pathogens was unknown. To do large microbial studies during commercial operation requires lots of planning and coordination. There are many factors to consider. Because the products are intended for human consumption, GMP and food safety compliance are absolutely critical. Because the experiments are taking place in a commercial setting, the logistics and cost are also very important. These considerations need to be balanced with the research needs, the result accuracy, relevance, and repeatability, and also disposal of research-related waste. But I'm happy to say we did it. With the strong support of our industry collaborator and several weeks of planning, our team moved to South Carolina. We converted conference rooms and offices into research labs and worked side by side with our industry collaborators. We conducted 9 research trials with 2 different process variations, so it is actually 18 trials total. We tested 3 different products, including diced cabbage, shredded iceberg lettuce, and chopped romaine. We took product and water samples every 30 minutes for up to 2 hours. We evaluated wash water organic load, pH, free and total chlorine, and total viable bacteria counts. We also collected production throughput, water, and chemical usage information. Here is the process flow diagram. We collected water from both primary and secondary flumes, or flume A and B, labeled here. For each flume, we collected water in 2 locations, the main flume and catch tank. This slide shows the dynamic changes in chemical oxygen demand, or COD, and pH when washing diced cabbages. On the left, you can see COD continually increased as more cabbages were processed. On the right, you can see the pH of the same wash water sample. The company did a great job in maintaining a relatively stable pH level during the entire process. Here are the changes in free and total chlorine. Free chlorine fluctuated during the process, while the total chlorine significantly increased over time. Bacteria counts in wash water of this trial was high early in the process, then dropped off very quickly and remained low for the rest of the trial. This may surprise you, but when you look at the free chlorine concentration, you will see why. In this particular trial, free chlorine actually went up. Keep in mind, the actual free chlorine concentration in wash water is the net balance of chlorine added and chlorine consumed, plus the water replenishment. Knowing that diced cabbage has a very high organic load and difficult to maintain the target chlorine level, the company certainly tried very hard. In this case, we ended up with increase in free chlorine towards the end of the process. These graphs are for chopped romaine lettuce. You can see the similar relationship between decreased bacteria counts and increased free chlorine concentration. After seeing that interesting trend, we plotted the relationship between free chlorine concentration and bacteria counts for all water samples collected during our diced cabbage trials. Here is what we found. Very high bacteria counts when the free chlorine was near depletion. With the increase in free chlorine, there was a substantial decline in bacteria counts. 90% of the positive bacteria samples we found had a chlorine concentration of less than 10 ppm. Here is the plot showing the data from the chopped romaine lettuce trials. We found much lower bacteria counts compared to diced cabbage wash water, but a very similar correlation between chlorine and bacteria. All but one, or 97%, of the samples that tested positive for bacteria had chlorine concentrations of less than 10 ppm. This is for shredded iceberg lettuce. Again, we see a very similar trend. With the help of our statistics specialist, we put data from all the commercial trials together. A clear trend emerged. With chlorine below 10 ppm and especially below 5 ppm, there was sharp increase in the number of samples that tested positive for bacteria above the detection limit. Above 10 ppm free chlorine, bacteria detection was sporadic with very small populations. Given the large sample size and the real-time detection during commercial operations, this finding is significant. Why so many weird signs? You probably are wondering. Well, just to make sure you are still awake, especially those international attendees who are trying to keep their eyes open in the middle night. But seriously, these signs are for things that we know, we don't know, and the action plans. First, what we do know. We have established the correlation between pathogen survival and cross-contamination with laboratory and pilot plant studies. We also established the association between the survival of native microflora and free chlorine concentration in wash water under commercial operating conditions. We know native microorganisms is a diverse group. It could include human pathogens, but also sporoformers that could be very resistant to chlorine, which may be attributable to those outliers that survive high chlorine concentration. Our previous study with tomato packing house dump tank wash water showed the shift in microbiome as impacted by free chlorine. However, research gap exists for fresh cut leafy greens. What we really need is the detailed comparison between real pathogens and native microflora in their response to different levels of free chlorine concentration. Simple studies with inoculation of overnight pathogen cultures are not very useful. What we really need are naturally contaminated pathogens that may have suffered cold stress, heat stress, nutrient stress, and are protected on plant surfaces. These conditions and data gaps are considered in our ongoing and future studies. Using generic E. coli, coliform, and Enterobacteriaceae as alternative to native microflora is also considered. Here is a list of our publications used to prepare this presentation. They are available, yes, for free on our website. Here are the key players on this project and our industry collaborators. Thank you all.
Thank you, Sunny. Our next speaker will be Dr. Shang-Hu Nu. He will talk about the microbiome dynamics on fresh produce in washwater and in processing environments and their relevance to produce safety. Shang-Hu?
Thank you. Now I talk about our work on microbiome. Microorganisms in any nature environment are present as multi-species communities. These microbial communities or microbiota plus the theater of activity constitute a microbiome, which is defined as characteristic microbial community occupying a reasonably well-defined habitat which has distinct physiochemical properties. However, many researchers, as is the case in our study, use those three terms more or less interchangeably. I'll briefly talk about our recent work on microbiome involving spinach, processing facility, and wash water. They are described in detail in our recent publications. For each study, I'll just mention one or two pieces of data and try to summarize the key points. First, spinach microbiome impacted by chlorine wash and storage. In this study, we used baby spinach from a collaborating commercial processor. The spinach was originated from growing areas in California and Arizona. Spinach were sampled before wash, immediately after wash, and packaging, and after 1 week storage at 4, 10, and 15 degrees. Total bacterial counts were determined by plating on TSA, and samples were treated with PMA before extracting DNA. PMA is a chemical that can effectively remove DNA from dead cells, so that only DNA from intact bacterial cells can be detected. The extracted DNA was used for cell quantification by qPCR and for 16S RNA gene sequencing to determine microbiome. This slide shows the bacterial counts on raw, washed, and stored spinach. Top is cell count by plating, and bottom 16S copy number count by qPCR. We can see that washing reduced bacterial counts by 1 to 2 log, and there was a significant increase in bacterial counts after storage at all temperatures. There are reasonably good correlation between the plating and the qPCR quantification. The impact of chlorine washing and storage on spinach microbiome was described at phylum and genus species level. This figure shows the relative abundance of identified genus or species. It can be seen that both chlorine wash and storage at different temperatures caused a significant shift of the microbiome. Some species, as those marked in red, were diminished by chlorine washing, but bounced back during storage. Some other species, as those marked in black, did not. Overall, the spinach microbiota after storage was more similar to that before washing. Similarly, this table shows that the species that had the greatest loss or gain in relative abundance after chlorine wash tend to head to the opposite direction during storage, which can be seen as an indication of the resilience of spinach microbiota. Now we summarize what we learned from this study. First, the microbiome on spinach is rich. We identified 644 OTOs, or operational taxonomic units. Then microbiome on the same commodity from different sources showed great similarity at a phenomenon level, but great variation at a genus species level. We also learned that chlorine washing diminishes, but not destroys, the microbial community. And the microbiota is resilient and tends to recover during the storage, even at low temperatures. Now I shift to our second study on Zone 3 microbiome. Food processors typically divide the facility surface into zones, with the zones 1 and 2 food contact surface and equipment, and zone 3 typically include and zone 3 typically including other surfaces inside the facility, such as the floor. This study was carried out at the processing facility of an industry collaborator. This slide lists the locations that were sampled, including floor, mezzanine stands, drain cover, entrance doors, and walls. We tried to cover different types of surfaces. Surface sponge web samples were taken before sanitation at the end of the day, and after sanitation before the morning shift. Before and after sanitation samples were from the same general location marked as adjacent squares. We took samples in both winter and summer. The samples were processed the same way as for spinach in the previous study. The total bacterial counts were determined by plating and by qPCR. We can see that the routine sanitation in general resulted in satisfactory cleaning for the surface on the production floor, including the floor, mezzanine stand, and drain covers. But the effectiveness on the peripheral surface, including the entrance door and the walls, are often questionable. This point marked by black is an exception, which was due to an unexplained flooding of the sampling site for one of the samplings. The microbiota relative abundance for the winter sampling is shown here. Although the total bacterial counts are generally low, there was a wide distribution of several species among the sampling sites. Interestingly, some species, as those marked by the red arrows, seemed more widely distributed among the sampled sites after sanitation, which suggested that the sanitation could have redistributed those bacteria. Now I summarize the key learning from this study. Again, the Zone 3 microbiome is rich. We identified over 8,000 OTEOs. We observed effective bacterial reduction on the production floor, but sanitation effect on peripheral surfaces, that means doors and walls, was less evident. We saw that effective sanitation resulted in both quantitative and qualitative shifts of Zone 3 microbiota. Several species were highly abundant at multiple sample sites for both winter and summer samples, and are tentatively assigned as core residential microbiota. Now we move on to the third study on washwater microbiome. In this study, baby spinach and Roman lettuce, with and without inoculation of foodborne pathogens, were washed in water to generate spinach and lettuce rinsate with the spinach or lettuce specific microbiota in the lab. The rinsate were then treated with different concentrations of free chlorine and PAA. The surviving bacterial counts and the microbiome were determined as previously described. This figure shows the counts of mesophilic aerobic bacteria from spinach and lettuce surviving free chlorine and PAA treatment. It can be seen that the survival of bacteria was strongly diminished at higher concentration of either free chlorine or PAA, with a higher reduction for spinach rinsite than for lettuce. Also, the inoculation did not change this trend. The washwater microbiota was dominated by a small number of genus or species, although nearly 3,000 OTUs were identified for species for spinach and lettuce combined. As the concentration of free chlorine increased, the presence of Pseudomonas and inoculated pathogens diminished, leaving Capria vidas with the most dominant presence. However, when treated with the increasing concentration of PAA, the inoculated foodborne pathogens remained high level presence, even they were not detectable by plating or enrichment. This shows that the cellular integrity of the inoculated pathogens were less affected by PAA than by free chlorine. This is the PCA, or principal coordinate analysis, showing the increasing similarity between spinach and lettuce rinsites microbiome at a higher concentration of sanitizers. The distinct spinach and lettuce microbiomes converge at the end at the highest concentration, at the highest sanitizer concentration, indicating the dominance of a common sanitizer-resistant microbiota. Here. Again, summarize the key learning from this study. Washwater microbial composition is diverse for spinach and Roman lettuce. Both chlorine and PAA significantly reduce the richness of washwater microbiota. At a higher concentration of sanitizers, washwater microbiome converges, indicating dominance by common resistant microbiota. Higher concentration of chlorine completely destroyed cell membrane integrity of the inoculated foodborne pathogens, making them not detectable by 16S sequencing. However, higher concentration of PAA did not destroy cell membrane integrity of the inoculated foodborne pathogens, allowing them being detected by 16S sequencing. By this far, many may already have questioned what is the use of the microbiome study for food safety. We have to say that microbiome studies are still at the early stage, and there is no consensus as how the microbiome information can be used for improving fresh produce safety. From the gut microbiome project, we learned that the microbiome are involved in many processes, which can also be true for other environments and processes, simply because the number and the diversity of microorganisms. It is critical to know what microorganisms are on the product or and in the processing facility which can help the understanding of many biological processes, including pathogen dynamics. Once we understand the microbiome on a product, in an environment or process, it can certainly be manipulated to our advantage to improve produce safety. We hope microbiome will not remain just as a concept. It will become a powerful tool for improving food safety and will be embraced by not only researchers, but also by fresh produce industry. Finally, let me acknowledge our team. Ganyu and Samantha for conducting the studies, and Dr. Otterson for helping with the DNA sequencing, and our industry collaborator for access to the facility. Thank you all for attending.
Thank you, Zhang, and thank you to all the speakers. Now we can entertain answering the questions. If you have questions, please enter them into the chat box that shows on the Zoom. I will begin to ask some questions here that came up earlier. Someone asked me about the depth of the tank water in the tomato wash studies. And those were lab bench studies that we conducted, and the containers that we use fit on our benches. So in some cases, the individual replicates were either 2 liters or 3 liters of water. These were sterilized before. They did not have any pressure variability objective incorporated into it. We did agitate during the commingling periods. So there was a period of stirring so that there would be an opportunity for contact between the different components.
Pat, the pathogen internalization studies for the tomato, we made sure that the tomatoes were submerged in the water no deeper than 2 layers of tomatoes, because I know that's we practice and we simulate.
Okay. We have a question here about the papaya. Someone wanted to know if the papaya that was used, was it from Mexico, and was it previously washed? Shango?
Yes. Yeah, the papaya, they were from Mexico. Simply at this season, I think in US, Mexico papaya is the only ones that are available. Yes, they are previously washed, and actually they are more, a little bit more ripened than those washed at Mexico, simply because importation, I think, importation process. That is one of the factors I mentioned. I say there are limitations about our study, and we cannot get the same Mexico as they do in, I mean, the same papaya as they do in Mexico. You know, it's generally are in a much earlier ripening stage, and ours were at a much later ripening stage, and it has been previously washed.
So let me add more. For the papayas, our earlier study, we purchased from the local wholesale market when we did the preliminary study. For the later studies, we worked with Jennifer and United Fresh and Dante and Texas produce. So we received donations from the industry, and those papayas were freshly harvested. For example, they were harvested on Sunday and then shipped to United States, of course, goes through the import process. As soon as the papayas arrived the US, especially in Maryland, we picked up and then we used either within the same day, if it's possible, or no later than the next day. So I want to take this opportunity to thank those people in the industry who donated us their papayas. Otherwise, they were expensive too, $3 each, and we used a lot. So thank you.
So, Shangwu, here's another question, but this goes to the talk that you gave on microbiomes. You talked about the number of OTUs, operational taxonomic units, but can you explain a little about what this really means? So we have some non-microbiologists here and people want to understand this better.
Okay, OTU, I think, as you said, is stand for Operational Taxonomic Unit. It basically means a group of microorganisms with the same characteristics. Not looking at a particular microorganism, just to see what common they have. But now especially means those different, when you do DNA sequencing, you have millions of sequences generated. All those sequences with a very similar sequence, they are called one OTU. By 16S sequencing, sometimes those OTU are at a phylum level, and sometimes it can be assigned to a species level. So it's a taxonomic term for DNA based on the DNA sequencing information.
Okay, we have a question for you, Sunny. This involves the studies with the tomatoes. Did the team track the time frame when you were sampling the flume? And if so, did you observe any bacterial counts when the water free chlorine content was less than 10 ppm in the water? And did that occur in the middle or the end of the run, or was that regardless of time?
So Pat, was that question related with my tomato presentation or the leafy green?
The tomato.
It says tomatoes. Okay.
The flume, yeah.
Okay, for the leafy green presented, we tracked the time. We measured every 30 minutes. For the tomatoes, actually, I think we were pretty random. We did not really track the time, but we did go in for cherry tomatoes. At one time, we tracked the time, but we did not see a huge difference in terms of the bacterial survival because the survival was very, very low. At one time, we did detect the positive bacterial counts. It was when there was a large load of tomatoes were dumped in the dump tank. and then you can see there was a sudden decline in the free chlorine. That was the time when what happened? Then that was the time we suddenly saw the increase in the bacterial counts. And that the counts in the dump tank was very low because they maintained the free chlorine concentration very well, far above what we had expected.
Overall, the study have some percentage of the number of samples that were less than 10 at 10 ppm free chlorine.
For the tomato study, because the company maintained the free chlorine very high, it was around 100 ppm. We barely detected anything most of the time. It was for the leafy greens because the chlorine concentration was much lower. When found the positives, most of the time were below 10 ppm, but the tomato, we really barely got any positives.
So there's another related question on how did you measure the free chlorine?
For the leafy green, we had the data from both sets. One is we measured the on-site using the DPD method. And we also received the internal data from our collaborator, which they used the very nice device to automatically tracking and dosing the pH and the acid. I believe it's called the FSIPC from Wash Solutions. And we were really happy with how the pH and the monitoring was done. For the tomatoes, we also used the DPD method for us, but there was no automatic measurement or record of pH.
So on the leafy green studies, how did you get the pH below the earlier recommended range?
For the pH, we did not do anything. It's the industry collaborator. They did very well with the pH control by using that FSIPC system. So it was automatically dosed the T128. Whenever the pH is high, then when they know they don't do anything. So it was maintained well by the company and of course in collaboration with that device developer.
Okay. I think we've covered all the questions. Does anyone have anything else that they want to submit as a discussion point?
Pat, I have a comment to make, actually two. First one, I know Dr. Xiang Wulong mentioned and thanked our collaborator Jennifer and Dangde from United Fresh and from Texas for the papaya work. Actually, there were a lot more collaborators involved in all of the studies we presented today. So I want to just give a shout out to all of those people. For our papaya study, we got the support, as mentioned earlier, from the importer and the people from the local wholesale market. We also received POAA from the chemical company. We actually requested one set of POAA, but we got 3 companies donated to us. So I really want to thank all of those POAA manufacturers who donated to us. For the tomato studies, which is another study that was requested specifically by the industry, we had a limited tomato proposal in our project. We did not have papayas, but because of the need for the industry to develop science and risk-based food safety practices, they need help to fill the critical data gaps. So they came to us. And then for the papayas, we contacted the USDA and IFA, got approval for expand our research. For tomato, it was limited, so we increased the scope. But I really also want to thank our employer, USDA ARS, the leadership teams at both the national and the local levels for their support and accommodation for our project team. And for the tomato one, we also got a lot of donations and unrestricted access from Suresh and Nipman produce. So I want to thank Suresh and Nipman produce. For the leafy greens, we also got a very strong support from McIntyre produce, allowed us to conduct research on their processing side. Earlier, we also got support from Smart Wash Solutions, conducted the pilot plant study that was presented. And we also got a very strong support from FDA. For the papaya study, we actually talked about moving our research team to Mexico to do this study, but eventually we couldn't do it because of the pandemic. But we got lots of support. So I just feel our studies really showcase the great collaboration and partnership between USDA NIFA, USDA ARS, FDA, and the industry. So I thank all of the people involved and the organizations involved. Without your support, we couldn't make this presentation today. So thanks.
Oh, very good. Thank you. We do have one other comment here, and this is for Zhang Wu. You talked about looking in Zone 3 for the microbiota studies. What was the limitation for Zone 1 and 2? The concern is that some people might think that they may have more relevance to food safety.
Yes, I would agree. But industry typically don't want you to touch Zone 1 and 2. So to get access to sample Zone 1 and 2, I think we'll need a lot more effort, coordination, and probably also we need more industry initiative. Rather, we try to ask for access. So it's just in general, it's not available, Zone 1 and 2 for our studies. So Zone 3, yes, I think most industry people even probably want limit access to Zone 3. But our collaborator, Sandy mentioned Taylor Farm. They are very great, very open, and they allow us access to all areas in Zone 3. We are very grateful to that. And also, I think Zone 3 microbiome is also very important for understanding the general microbiome in the process, in the processing environment. So Zone 1 and 2 will say are more relevant, maybe, but generally you don't find the rich microbiome as you can find in Zone 3.
Do you want to talk at all about how you all neutralized the chlorine when you did the microbial analyses over all these different studies? Do you want to address that, Sunny?
Okay.
Yeah.
Actually, we have a specific session this afternoon presentation about the neutralizer, but just a quick answer. We use the sodium thiosulfate to neutralize. But
Okay.
it's very important you have a sufficient amount. So we'll talk about the details this afternoon.
So there's a question about will these presentations be recorded? Yes, they are being recorded, and these will be put on our project website. And I think we'll get to a point of showing that website in forthcoming.
Yeah. It's also on my opening remark, our project website. If you want to write down, it's producefoodSafety.org.
So we'll be sure and show that again, and we'll post it on the chat as well.
Yeah.
Are there any other questions or conversations, discussions at this time? Okay, Dan has just posted the project website, so it's in the chat. You can see that. Well, if not, we will conclude this session, and thank you all for your participation and the lively discussion. And we will join back again for the next session, which will be at 2 o'clock.
No and no. Yeah, the pH is high, I'm sorry. They dose the acid into the flume. With very important set of data to everybody.
USDA is an equal opportunity provider, employer, and lender.